Good day, students. I would like to welcome you to study session 9 of NSA 307, which will discuss digestion and absorption of food. Please sit down and relax to enjoy this lecture. Foods that animals eat and even human beings goes through four main processes, which is known as ingestion, digestion, absorption, and defecation. Upon ingestion of food, digestion happens first, and absorption of nutrients in digested food takes place next to generate energy. These processes takes place in the alimentary tract of an animal. These two processes are completely different processes. However, it is impossible for absorption to take place without digestion. So in this study session, you'll be introduced to the process of digestion and absorption. At the end of this study session, you should be able to explain digestion and absorption, to include their differences. You should be able to describe the processes of digestion and absorption, and also you should be able to discuss the phases of gastrointestinal control. Let's look at digestion. Digestion is generally the breaking down of food containing processes or series of processes, and there are two main steps of digestion, which is known as mechanical and chemical. In digestion, simplification of large molecules into small monomers takes place. That is, it is a process of catabolism. However, there are mainly two forms of digestive systems. The primitive organisms have external digestion, while more evolved advanced animals have internal digestive systems. In advanced animals, digestion starts in the mouth and continues through to the stomach and concludes at the jejunum. While food passes through esophagus, the peristaltic movement helps to break it down into smaller particles. Inside the stomach, the chemical digestion becomes predominant with secretion of digestive enzymes and acid with optimum temperature. There are 6 processes involved in digestion. It includes 1. The movement of food and liquids. 2. The lubrication of food with bodily secretions. 3. The mechanical breakdown of carbohydrates, fats, and proteins. 4. The reabsorption of nutrients, especially water. 5. The 6th one, the production of nutrients such as vitamin K and biotin by friendly bacteria. And lastly, the excretion of waste products. Comprehension of the tasks of processes needed to break down food is essential to an understanding of how and when food really begins to function within the body. For example, not understanding that carbohydrate breaks down into glucose could lead one to believe that the best source of glucose is in the liquid form, such as a soft drink. This could cause one to miss out on the nutrients in fruits, vegetables, and grains. Likewise, not understanding the digestion process could lead a person to believe in the might of food combining, or perhaps to think it is normal to be hungry all the time. But in fact, the digestive processes normal to human physiology can simultaneously handle carbohydrates, fats, and proteins and allow people to go several hours between meals, especially if meals are balanced in fiber and the individual nutrients needed. Let's look at the process of absorption. It's a transferring the digested molecules across the gastrointestinal tract into the bloodstream. Absorption will start in the stomach. It continues through the small intestine and concludes at the large intestine. Absorption has to do with the movement of molecules across the gastrointestinal tract into the circulatory system. Most of the end products of digestion, along with vitamins, minerals, and water, are absorbed in the small intestinal lumen by 4 mechanisms for absorption, which includes active transport, which requires energy, passive diffusion, endocytosis, and facilitative diffusion. Nutrient absorption is efficient because the gastrointestinal tract is folded with several surfaces for absorption, and these surfaces are lined with villi and microvilli cells. Efficient absorption can be compromised due to lactose intolerance. It is usually due to the lack or absence of the enzymes lactase, which breaks down milk sugar. Protein, carbohydrate, lipid, and most vitamin absorption occur in the small intestine. Once proteins are broken down by proteases, they are absorbed as dipeptides, tripeptides, and individual amino acids. Carbohydrates, including both sugar and starch molecules, are broken down by the enzymes in the intestine to disaccharides called sucrose, lactose, and maltose, and then finally into the end products known as glucose, fructose, and galactose, which are absorbed mostly by active transport. Lipase is an enzyme in the pancreas and the small intestine and bile from the liver. They break down lipids into fatty acids and monoglycerides. These end products are then absorbed through the villi cells as triglycerides. Alcohol is not a nutrient, but 80% of consumed alcohol is absorbed in the small intestine. The other 20% is absorbed into the stomach. Alcohol is absorbed by simple diffusion, which explains why gastric ulcers are not uncommon in people who drink excessively. Jejunum and helium, they absorb most of the nutrients, while the large intestine does majority of the water absorption. And finally, absorption at the large intestine, the undigested and unabsorbed portion is ready for defecation. Looking at the differences between digestion and absorption. Digestion is a mechanical and chemical breakdown of large food contents into smaller particles that are easy to be absorbed. Absorption is transferring the digested molecules across the gastrointestinal tract to the bloodstream. Digestion will start from the mouth, but the absorption starts from the stomach. Digestion takes place inside the intestinal tract, but absorption happens in the line of the intestine. Digestion needs enzymes, but absorption does not need enzymes. Digestion is always an active process and needs energy, but some of the absorption mechanisms do not need energy. Digestion changes the chemical and the mechanical structure of the food, whereas absorption does not. Absorption relates with blood, while digestion does not. Also, most of the digestion takes place at the duodenum, whereas absorption occurs mainly at the ileum and the jejunum. Absorption operates on both simple molecules of digestive and non-digestive matter, while digestion functions upon large and complex digestive matter only. The human physiology of digestion and absorption of food. The gastrointestinal system includes the gastrointestinal tract, which comprises of the mouth, the pharynx, the esophagus, the stomach, the small intestine, and the large intestine, and also other accessory organs like the salivary gland, the liver, the gallbladder, and the pancreas that secrete substances into the tract via connecting ducts. The gastrointestinal system breaks down ingested food into molecular forms by enzymes, which is the process of digestion, that are then transferred to the internal environment through absorption. Looking at the functions of the gastrointestinal organs. The gastrointestinal tract begins at the mouth, where digestion begins with chewing. Saliva contains mucus. The enzyme amylase is secreted from 3 pairs of salivary gland, which is located in the head. The mucus moistens the food, and amylase partially digests polysaccharides, which are starches. Food then reaches the stomach through the pharynx and the esophagus. The stomach is a sac that stores and digests the food macromolecules into a solution called the chyme. Glands lining the stomach secrete hydrochloric acid that dissolve food particles and protein digesting enzymes called pepsin. Final stages of digestion and most of the nutrient absorption occur in the next portion of the tract, which is the small intestine. The small intestine is divided into 3 segments. We have the duodenum, the jejunum, and the ileum. The pancreas is a gland that is located behind the stomach. From its exocrine portion, it secretes digestive enzymes and a fluid that is rich in HCO3 ions to neutralize the acid from the stomach. The liver secretes bile, and bile contains ions and bile salts to solubilize fat. Bile reaches the gallbladder through hepatic duct and is stored in the gallbladder between meals. During a meal, bile is secreted from the gland by smooth muscles contraction and reaches the duodenum portion of the small intestine by the common bile ducts. Monosaccharides, amino acids, and mineral salts are absorbed by transporter-mediated processes, while fatty acid water diffuses passively. Undigested materials is passed to large intestine, where it is temporarily stored and concentrated by reabsorption of salt and water. And finally, contractions of the rectum, which is the last part of the intestine, expel the feces through the anus through a process of defecation. Looking at the structure of the GIT wall, the luminal surface is covered by a single layer of epithelium containing exocrine and the endocrine cells. The exocrine cells disintegrate and discharge into the lumen, releasing their enzymes. The epithelial with an underlying cover of connective tissue, that is the lamina propria, and the muscle, which is the muscularis mucosa, are called mucosa. Below the mucosa is a layer of the inner circular and outer longitudinal smooth muscles called the muscularis externa, which provides the forces for moving and mixing the GI contents. The outermost layer of the tube is made up of connective tissue called serosa. The luminal surface of the tube is highly convoluted into projections called the villi and the microvilli, both of which increase total surface area for absorption. The center of each villus has a single blunt-ended lymphatic vessels called the lacteal, and there's venous drainage from the intestine transport absorbed materials to the liver for processing via the hepatic portal vein. Then also the process of digestion and absorption in classes of nutrients. In carbohydrates, digestion begins in the mouth by salivary amylase and completed in the small intestine by the pancreas amylase. Monosaccharides such as glucose, galactose, and fructose are produced by the breakdown of polysaccharides and are transported to the intestinal epithelium by facilitated diffusion or active transport. Facilitated diffusion moves the sugar to the bloodstream. Also, in proteins are broken down to peptide fragments by pepsin in the stomach and by pancreatic trypsin and chymotrypsin in the small intestine. These fragments are then digested to free amino acids by carboxypeptidase from the pancreas and amino peptidase from the intestinal epithelium. Free amino acids enter the epithelium by secondary active transport and they leave it by facilitated diffusion. The small amount of intact proteins can enter interstitial fluid by endo and exocytosis. Also, looking at the process of digestion of fat. Fat digestion will occur by pancreatic lipase in the small intestine. A monoglyceride and two fatty acids are produced in the digestive processes. Also, large lipid droplets are first broken into smaller droplets by a process called emulsification. And emulsification is driven by mechanical disruption and emulsifying agents. Pancreatic colipase binds the water-soluble lipase to the lipid substrate. Digested products are bysold from the amphitaphic micelles. These micelles keep the insoluble products in insoluble aggregates from which small amounts are released and absorbed by the epithelial cells via diffusion. Free fatty acids and monoglycerides then recombine into triacylglycerol at the smooth muscles endoplasmic reticulum and are processed further in the Golgi and enter the interstitial fluid as droplets. Looking at the digestion and absorption of vitamins, fat-soluble vitamins are absorbed and stored along with fats, while water-soluble vitamins are absorbed by diffusion or mediated transport. Vitamin B12, because of its large size and charged nature, first binds to a protein called intrinsic factor, which is secreted by the stomach epithelium and is then absorbed by endocytosis. Water, the stomach absorbs some water, but most is absorbed at the small intestine by the process of diffusion. The phases of gastrointestinal control. Each phase is named according to where the receptor for a reflex is located, and these phases do not occur in temporal sequence. The cephalic phase is initiated when you sight, you smell, you taste, you chew, and even with emotional states, they stimulate receptors in the head. Reflexes mediated by sympathetic and parasympathetic fibers activate sensory and contractile activity. At the gastric phase, it is initiated by distension, acidity, and the presence of amino acids and peptides in the stomach. And this phase is mediated by short and long reflexes and activates the secretion of gastrin. And the last phase, which is the intestinal phase, is initiated by distension, acidity, osmolality of digestive products in the intestine and is mediated by gastrointestinal tract hormones. It is short and long neural reflexes. Thank you for listening. See you in the next slide. Bye for now.